OmicsLogic is a place where you can learn by doing. We combine theoretical knowledge and understanding of biomedical data and bioinformatics with practical examples where you can apply what you have learned and grow in your ability to start thinking creatively, develop independent projects, and develop the skills that you need to really be able to be proficient with bioinformatics. This can be done in either one of two ways. You can either focus on the biomedical research to develop research and curiosity using data analysis, or you can be more focused on developing coding skills and analytical thinking. These two tracks are divided by the type of tool that we're going to prioritize throughout the learning. If we're focused on the biomedical research side, we're going to primarily rely on a user-friendly interactive tool called the T-Bio Informatics Platform. There's little to no coding involved. You primarily assemble together pipelines or sequences of steps to understand the outcomes. On the other hand, if you're more focused on the data science or the skill-based approach, you're going to prioritize learning syntax, different packages, and analytical methods implemented in coding languages like R and Python. So as you develop those coding skills, you will also be introduced to different types of challenges and projects, but primarily that illustrate how specific challenges are addressed using skills in coding. First of all, let's take a look at how this could be broken down in a real program. So let's say you are interested in genomic data. Genomic data can be illustrated with a variety of different projects. One such example is SARS-CoV-2, where we compare sequences of the SARS-CoV-2 virus and we study how it developed over time, accumulating mutations that led to different types of outcomes. As you learn about what genomic data looks like, you are then prepared to analyze genomic data. The idea is to have enough understanding of what that data looks like to be able to perform analysis and interpret it biologically. So what would be the outcome? As the outcome, the student should be able to show differences between multiple pathogenic strains and show how maybe those map to the protein structure of the virus that is responsible for the viral entry into a cell. Let's say you're interested in transcriptomic data, RNA-seq. Then you can take a project called the Cancer Genome Atlas Liver Cancer Project, where you will again understand the context. What is cancer? What is gene expression? How to compare sets of genes that are differentially expressed. Then you will take that data set from the Cancer Genome Atlas that has patient data about many different types of cancers and run a pipeline, which will produce an interactive dashboard that will allow you to explore different genes, pathways. And so as the outcome, the student will identify differences between cancer types or between risk cancers or between risk factors that lead to liver cancer, for example, hepatitis infection or alcohol consumption, and explain gene function responsible for those differences. The practical side of that program is essentially around activities that connect different analysis steps together in what we call an analytical pipeline, and to identify the right set of steps for the right kind of data that answer a specific research question. As a result, the interactive outputs make it easy to explore different types of visuals and elements like genes, their expression, etc., which are commonly used in publications that kind of explain the research that is being done. At the end of that, the student potentially can develop an independent research project. Now, developing a research project independently takes time. So the first step is preparation and giving an example. And then after the person is comfortable, understands the analysis, can work independently, and has a reasonable project proposal, they can start developing that independent project. Before they do that, we have a way to track their activity by assigning points to different steps that they perform and giving them program badges when they interact with mentors around a specific domain that they are most interested in. Now, some students decide not to work on an independent project, and that's why we provide them with curated data sets and initial steps that can help them get started with a project across many different types of data. For example, cancer macrophages, modeling cancer precision medicine, the tumor microenvironment, malaria infection, potato resistant, drought resistance, and the liver cancer, and the Cancer Genome Atlas for liver cancer risk. On the other hand, some students want to develop an independent research project. Those kinds of students are motivated primarily by working on an independent project, and they need the technical support and the tools to be able to identify data, collect it, and organize it the way that they need to, and then perform the full analysis with an emphasis on biological interpretation. Here's an example of a high school student who has been a part of a program designed by the Science Coach organization, and the outcome was that she was able to present this research project at a science fair. When she presents that at the science fair, she's able to demonstrate that she was able to address a research question and interpret the research findings that she herself went through and analyzed. Now, some students are going to be more focused on just completing coursework and having skills that they feel enhance their profile. For example, this student at the Georgetown University Medical School wanted to acquire skills in machine learning applied to biomedical data. Others could be even faculty members, like you see here, faculty members that are learning bioinformatics for the first time, or international students that come from a variety of backgrounds and learn throughout the course to just establish their understanding. Now, if we talk about the data science or the skill-based approach, here the idea is a little bit different. Obviously, the same idea of developing a project or using data to apply their analytical thinking applies, but the focus is more on developing coding skills to analyze biomedical data. And the outcome would be to demonstrate that they have proficiency, they understand the syntax, they know how to write code, they know how to make the code more efficient, and then they present what they have done using those coding skills to a mentor that can provide them with feedback. So a lot more emphasis here on understanding syntax of either R or Python, and using standard packages for data wrangling, visualization, statistical analysis, and machine learning. Again, different types of data could be used to do that. And so the choice on what kind of data can actually dictate what kind of exercises they will perform. At the end, the student will develop a good understanding of how to take different parts of code and organize them into a Jupyter notebook, to understand the syntax and be able to edit that or maybe even write code themselves, and perform coding challenges where they can test how well they understand what they have done and essentially produce an application or an analysis notebook that they can demonstrate to others. Because coding is something that many people learn, we also recommend developing a GitHub profile, as this student from the Georgetown Medical School did, where you can submit a repository of developed code, and that essentially introduces you to the community of developers and data scientists that are on that platform. And you are using examples from the projects that you did throughout the program to attach that to your outcome and to your resume as well.